and management, you please consider the time. The time is very important for research. You have to allocate time for the faculty member who can do the research and who can engage in the research, what they can do, provide you output. By the same time, I also mentioned to all the young teachers, and the research is not a 9 to 5 job. You also don't think that the research could be done 9 to 5 job, and you have to dedicate your time irrespective of a 9 to 5 job, and you have to dedicate yourself in the beginning to reach a milestone. So this is one of the analogy I always quote to the, like, when you go for your education system, it is an analogy like, there is a road, there is a school van on the road, there is a school children's, the school driver, and how you are going to compare. So now the road is a program, like a B-Farm program. The van is a curriculum, the B-Farm syllabi, which is prescribed by Pharmacy Council of India. The school children are students, and the instructor is driver. See the role of teacher. That doesn't matter, the program road is good, van is good, students are good. If your teacher is going to be wrong, the things will collapse. Similarly, I wanted to say that, the school engine, van engine is the administration system and management. It is very essential that engine of the car or engine of the van should be perfect enough to drive, to take the smooth educations. This is the best analogy whichever I want to convey to the complete education system. And another challenges for academician, right? So as of now, the world required, our nation required to minimize the gap between academia and industry. So we are supposed to decrease the gap between academia and industry. That is the reason now institutes are planning to now, even for this conference, and I'm happy that you invited some people from industry to address. And so here, the reason nothing but why in India there is a huge gap between industry and academia, and we have different mindset, different goals, perspectives difference, cost and aspiration. The reason exactly that, the one major thing I wanted to point out here, the cost matter. The industry perspective cost is, they always have very, it's a kind of, they always think economically. But in the case of your academic research, we don't think about the cost, we always think about the research output. But when you want to integrate your academic research to industrial research, and you are supposed to integrate the cost effectiveness as well as the research output. That is one of the major challenging ahead. And when it comes to is, I don't know how to this minimize this. Is my screen is completely visible or any obstructions?
No obstructions, sir.
Okay, okay. In my screen, something is visible. So, okay, it's fine. No, it's completely clear, right?
Oh, yes, sir. Yes, sir.
Okay, sir. Okay, okay, then. Okay, fine. Okay. So now, this slide, it may be a surprise to you. I don't know. Can anyone guess that how many human diseases are exist in the world? I'm sure there are about 1 lakh human diseases have been identified. But out of 1 lakh human diseases, there are 7,000 diseases are recognized as rare diseases, where the mechanism is not known, target is not known, treatment is not available. So it is a more, it's a kind of tonic. This slide can act as a tonic or some kind of act as an inspiration to all the researchers that and people who are involved in drug discovery. If you consider the current rate of drug development, and we require another 500 years to get a treatment for all these conditions. So really it is a tonic or a kind of inspiration to all the people who are in the drug discovery. So now when I speak about drug discovery, why we do discovery? To treat diseases. So when you want to treat diseases, the question is comes that what diseases has to be or have to be chosen. So now I shall categorize these diseases into two categories. One is infectious disease, another is non-infectious disease. And you are aware of that infectious most dangerous as of now are smallpox, plague, like MDR and XDR tuberculosis, malaria, dengue, Ebola, COVID, cholera, etc. And these are the things more commonly, happening with this particular infectious disease. When it comes for non-infectious, like heart diseases and even the drug-resistant cancer and chronic lower respiratory disease, which is not related to infections, and cerebrovascular diseases, Alzheimer's and diabetes. So now, and if we can categorize the deadly diseases, one side is infectious, another side is non-infectious. What our society needs now, which area you are to focus, which is the prime important. Now the case is here, it is unfortunate that drug resistance infects diseases, which ultimately need new drugs. For example, are you aware of that Staphylococcus aureus infections? More commonly, we treat with the penicillin. Then the Staphylococcus aureus developed resistance with the methicillin that become MRSA. So we used vancomycin to treat MRSA, and over the period, even the vancomycin also become resistance. Now it become BRSA. Similarly, tuberculosis, rifampicin and isoniazid was the most important sterile drug once upon a time. But as of now, no. Alone this pyrazinamide, isoniazid, ethambutol, rifampicin, and they are not at all working at all. Now that the treatment of tuberculosis is very complex and they are all even becoming cardiotoxic. Despite the toxicity, even the treatment success rate is only 50%. People are dying. The death rates are increasing because of the drug resistance. So here we need that new drug molecule. If we come to the other side, you know that there are, people with diabetic, heart diseases, neuro diseases. We have polypharmacy. Once upon a time, the prescription size was a small paper, very small paper. It is just a piece of paper. Now the prescriptions are A4 size paper. You are aware of that. And all because of, the doctor not able to accommodate a multiple medication in a piece of paper. Now we become A4 size paper. A polypharmacy. And because of that, ADR, adverse drug reactions, toxicity, that is a need that created a huge job for PharmD people, pharmacovigilance, ADR monitoring. So now for this area, we need milestone research. The milestone research, a kind of research where you can put a full stop. It is not a managerial, you need a curative, but it's very challenging. Even in near future also, it is not possible. So now here I'm going to talk, I just wanted to insist more of antibiotic resistance. It is the biggest, it is an invisible war. We speak about First World War, Second World War, and we also, many people are thinking about, Third World War. So that World War can be concluded. It is in our hand. But this war, biggest fight between your microbes and our drug, now almost, the microbes won the war. And almost all microbes developed the resistance to the existing antibiotics. So one fine day, the antibiotic therapy going to be a placebo therapy, definitely. So it is a need for new drug and new antimicrobial or new antibiotic, not only to bacteria. I want to insist it is also to fungi. We developed drug resistant fungal candidiasis, drug resistant malaria, drug resistant viral infection diseases, drug resistant tuberculosis, drug resistant leishmaniasis. So, and so what? Every microbial kingdom, bacteria, fungi develop resistance in a very faster rate. It's very faster rate. So you can see this. So this is a kind of review on antimicrobial resistance. It is a expected and predicted deaths by 2050. You can see that out of all the death due to the antimicrobial resistance, it may cause 10 million deaths by 2050, followed by cancer. It is expected that 8.2 million deaths are expected in 2050 for cancer, 10 million deaths are for infectious disease, followed by next is diabetic 1.5 million, diarrheal diseases, road traffic accidents, and measles, cholera, and tetanus, etc., etc. So now you can see this slide, this expectation, the prediction that speaks about how dangerous this antimicrobial resistance and what is the need of new antibiotics, new antimicrobial in very new future. Can you see this slide? I think it's very minute to see. It's, I think, just like taken from the report of CDS. And this is the kind of two report. The report shows that in 2013 and 2019 about how many pathogens are developed resistance and how many strains of drug resistant strains are existing. So exclusive.
Ramlingam, sir.
Yeah.
Sir, sorry for interruption, sir. Sir, the slide isn't moving, sir.
Okay.
Sir.
Now?
Oh, no, sir.
Okay, just check.
Change up. 60 slide back.
Now?
Yes, sir. Now we can see.
Okay, now it's changing, right?
Yes, sir. It is changing.
Okay, then. Okay, sorry for the interruption, maybe due to the technical error. Okay, so when you come for this report 2013 and 19, and you can clearly see that in 2019 report, and you can see that, you can see that assault resistant aspergillus, drug resistant mycoplasma, drug resistant, and Bordetella. So here also you can see that in constant threatening also, and there is, and we have developed so many, there are so many organisms are developed resistance, and in case of this, right? So only one thing, right? If you look at, and the vancomycin resistant Staphylococcus aureus VRSA, it is not existing now, and remaining all is added now. You can see here, and earlier there was no carbapenem resistant Candida aureus. So now it's also, the developed, drug resistance Candida aureus has developed in that become the urgent threat now. So here you can see the list. If you read out, just simply a key message, what you understand from the slide, and most of the pathogens are resistant now. No, like earlier we speak only the Acinetobacter, but now we call it as a carbapenem resistant Acinetobacter. And we never say, we go for necessary as a gonorrhea, we study in pharmacology book. Now there people are calling it a drug resistant necessary as a gonorrhea. So similarly, Staphylococcus aureus, we call it as methicillin resistant Staphylococcus aureus. And Enterococcus is one of the most dangerous. But now the vancomycin was given. Though vancomycin is a toxic antibiotic, we used to treat with vancomycin. Now this particular Enterococcus also developed resistant towards vancomycin. So coming to this classification of drug resistance, it is just a fundamental, and people who are new, just a 1 minute glance to the people. And as of now, based on the various reports, the research on how a microbe develop antimicrobial resistance to a particular antibiotic. They categorized three different category. One is intrinsically developed, AMR. Another is adaptive based AMR. Another is acquired AMR. The first category, intrinsically developed AMR, it's something like it's purely due to the low permeability drug to walls. So some drugs cannot pass the cell wall membrane because this drug resistant microbes developed cell wall thickness. And example, these aminoglycosides, and it can sometimes this gram negative bacteria, it's develop or modify the cell wall such a way that this aminoglycoside cannot cross the cell membrane. When it comes for adaptive based AMR, and it is purely due to the chemical stress. Sometimes, and it is because of the environment trigger. Continuously, when a particular microbes or pathogen is exposed to a mild and subtherapeutic level of antibiotic concentration, which is the less than the MIC. So this, because of the low MIC, and it cannot kill, but what happened, it start adapting this particular when in terms of the change in the gene, and so that it develops some intermediate, it produces some intermediate and metabolite in the particular microbes, and which can able to tolerate it. And third one we call it as acquired AMR. It is acquired nothing but purely because of the gene transfer, where the particular gene from one particular microbes is transferred to another microbes, and where this kind of gene transfer mechanisms, and automatically this starts, the acquired AMR starts. So now the question is here, when it comes for drug discovery, and now we are the era of the third one, because acquired AMR is very concerned. Acquired AMR is very concerned now, and adaptive AMR, acquired AMR, because if you feel that your microbes develop only change in the cell wall, or the microbes change in the kind of efflux pump, where you can able to target, we can design alternative medicine or alternative antibiotic or derivative antibiotic. So the derivative of antibiotics works only when this particular microbes have intrinsically developed the AMR. But here in the acquired, it's almost like, most of the more than 80% of the microbes are in these two category only, adaptive based AMR and acquired based AMR. That is the reason why I'm saying that, if you look at your WHO or FDA or our thing, as of now, we don't have any new drugs or alternative drug to penicillin. There is no alternative for aminoglycoside. Nothing is alternative for other class of antibiotic. And they're trying to introduce this antimicrobial, antimicrobial stewardships, like a kind of program. They're trying to, avoid and minimize the irrational use of antibiotics, and they're trying to educate the knowledge. Educate the people, educate the doctor, health sector, and all those things are people are doing it now from the clinical sector and the ICMR or whatever the case. They're trying to do it because no other case they have to do that. But in fact, in reality background, imagine that if you go to the clinical trials now, the clinical trials are now with the 5 combination of antibiotics, 3 combination of antibiotics, 4 combination of antibiotics. Still, the drug resistant species are not responding. It means, and these microbes have transferred from, drug resistant to MDR, multi-drug resistance. So the question here, nothing but drug resistant may be like, your microbes is developed resistant to only one class of antibiotic. So where you can choose other class of other mechanism antibiotics. But the question is here, right, when the drug become MDR, multi-drug resistant microbes, and even, it has developed resistance, it developed adaptive antimicrobial resistance, or in terms of your gene transfer to almost all kind of antibiotics. The realistic background, the existing antibiotics or the existing drug molecule, the existing mechanism will never works at the MDR strains. So at the ultimate, there is the ultimate need of a novel mechanism. That is the need of the hour and need of the day for that today's drug discovery against drug resistance. And this slide, if you see, and you can see it is a kind of the extractive data of FDA. The orange color speaks about the cancer drug, and the blue color speaks about, antibiotic. It is just a short span of time from up to 2014, a kind of data from the nature. And the question is here, I believe it's my personal opinion or point that, and it is the negligence of our researchers and who forget to take up the antibiotic drug discovery at right time. Because this drug resistance, it is started in the era of 1990 only. You are aware of that. The simple, Staphylococcus aureus is developed resistance, against penicillin 1980 itself. It is a concept that drug resistance is evolved in around 30-40 years ago itself. But only thing is that because of the other antibiotic availability, we are not given concern about the alternative, or you are not given concern about, subsiding the resistance. Now almost it is reached the threshold. There were nothing to now. There is no strategy or no other stewardship program can subside the resistant development. It is already developed. Now that is a need for going for alternative to, compact the resistance now. So we cannot use any subsiding the resistant mechanism. We cannot go for any subsiding strategy at all. It is a need to compact the resistance and to find a, missile to target the particular drug MDR strain. So that's what here it is also a fault of pharma industry also, right? People are not given concern about this. It is very true that, most of 80% of pharma company, they go towards your business motto than the health motto. So you can see that, the people felt that, cancer is used, they get so much money from cancer medicine. You can see that is a tremendous growth of, anti-cancer drug discovery attempt by industry and approvals when compared to antibiotic. You can see antibiotic drug discovery in 1980, it was very high. Next, exactly after 25 years, it is the antibiotic drug discovery is declined to 25%. Whereas in 1980, it was only 5%, and it has grown up to 100%, more than 100% in 2014. So it was a complete, it is both, antibiotic drug discovery, anti-cancer drug discovery, totally in reverse in this scenario. And once again, this is a kind of, and where you can see that how this pipeline drug. So this graph shows about your, the percentage or the statistics of the different drugs in phase 1, phase 2, phase 3, and phase 4. You can see that the orange color, you can see this orange color. This is the new drug applications. In 2009, it is relatively very low. But when you come for even now, you just look at that, the phase 1 drug, phase 2 drug, it is not so enormous. I mean, it is not, so significantly considerable number at all. Okay, so here, even when you go for a green line, the pipeline drug, and in 2014, from 2014, it gradually increased. You can find something about 2017 very high. Once again, it is subsided in the recent years. So here, why this series? I am telling that there is a subsiding. We cannot expect that a significant number of molecule that is not enter into the clinical trial. That is a major challenge now. It doesn't. I don't blame that the researchers are not doing antimicrobial research. Throughout the world, many scientists from the academia, industry, people are doing drug discovery. They are synthesizing new molecule. They're screening it. But some of the other limitations, and it is not entering into the pipeline of phase 3, or is not coming into your clinical trial also. The reason, nothing but the major challenges associated with the thing, but it's called as de-risking at early stage therapeutic targets. It's very important. It's very, very important that a scientist or who work on this particular antimicrobial be required to de-risk at early stage of therapeutic target. So the de-risking, nothing but you require to have a integrated attempt. You have to identify a drug target. You have to validate it appropriately by interdisciplinary scientists. So one, you should know about the gene editing technology where you should be able to know that what gene is responsible for the resistant, what is the gene expression profile, and whether it is particular target is really reliable for the drug resistance, and how whether the particular target is having a very low potential for further mutations, and you have to prove with your animal model, and you have to see that molecule is stable or not. In molecular chemistry, biological, there are some multiple disciplinary has to be involved at the initial stage of screening and drug target identification validation, and that's the reason, and we are supposed to do that de-risking at the early stage therapeutic target. So another challenge is, we speak nothing, but it is very essential that, and we are well aware of a human biology, human anatomy, human physiology. We are aware of it. We have plenty of textbooks, plenty of things have done. But when it comes for the cell biology, it is very difficult to understand the cell biology of an MDR strain, cell biology of your XTR strain. So it's very example, and even now, it's very example. It is very difficult to differentiate MDR TB and XTR DB, multi-drug resistant tuberculosis and extensively drug resistant tuberculosis. It's both. It's very difficult to. Differentiate these both. It's very difficult to differentiate these both. The reason nothing but, and we are supposed to understand what is the signal transmission mechanism, growth regulation, and how these particular microbes adapt and how it respond to the environment. Once again, these are defense and synergy mechanisms and communications and proliferations, and it's all the parameter which are interlinked to the antibiotic drug discovery. So until and unless if you are able to elucidate the things in your particular drug resistance microbes, and then only it is possible to go for your de-risking and kind of you can try to identify the exact drug target. So now it's very important component, and it's my recommendation. It is my recommendation, or I can take it as my advice to all the people who are trying to do the antimicrobial drug discovery, and you try to have knowledge on the gene expression profiling of the particular microbes. So because until and unless you have the gene expression profile of the microbes, and it is very difficult to examine, right? So this particular gene expression profile can help you, to examine the basal, like, to examine the cells, and this can help you out to identify the gene expression and signature, and it will reveal the metabolic pathway of a drug particular cancer or microbes, and this will help you to identify the new target for drug design. And also, it's very important that if you're aware of the gene expression profile of a particular microbe, and it can easily to predict the clinical behavior, how these particular microbes will behave in a human in infectious conditions, whether the particular molecule can be given to treat the particular treat the infections or microbes in the condition or not. This all can be elucidated, and where you're required to have your gene expression profile. The gene expression profile, nothing but as of now we have proteomic studies, metabolomic studies, and we have here gel electrophoresis SDH page, and it's all like a microarray techniques. There are so many things are there, the molecular biology tools, which is very essential as of now. The conventional way of synthesizing just a screening, and it's only the preliminary, it doesn't work. And these are, there are some questions to be addressed in drug discovery for, drug target discovery, especially in drug target discovery. So what questions there are? Almost like 13 questions to be assayed. So like it's all a very fundamental questions only, like the target. It's relevant to the human cells or tissues, where the target is associated with the clinical condition or not. What is the genetic correlation of the target? Whether the target should be blocked or activated, or is it possible to practically feasible or not, and validation is possible or not, and whether the small molecule or antibiotic can target or not. is it possible to, do some kind of in vivo model or not? And how well does it model predict the human disease? And the target system, the target inhibition or activation can be assayed or not. So likewise, is all these questions has to be addressed before you try to identify a target for antimicrobial resistance. So once again here, there are some plenty of gaps, the gaps in the current drug discovery. The reason, nothing but there is a disconnection between your early and late drug discovery. That is a major gap in the current drug discovery. So you know that the early drug discovery is purely focused on the genetic validations, and a major predictor of the success. But still, if you're, as of now, we say that gene knockout and gene knock-in techniques are one of the important drug validation tool as of now we are using in mouse, but still there are simplistic way. So that is the case, and as of now, it is very, very important that when you identify a drug target, for example, in your microbes, you're saying this is a target, and what we should have a facility, we should have a base that we should go ahead with your knockout. You develop your knockout microbes, and then you screen a compound, and then you say that the particular, we can have your two output from that. When you try to screen your compound and the knockout microbes, you will come to know whether your molecule is exactly acting on the particular target, or other than that, it's acting on other target or not. Second, how far the survival potential is existing in the particular microbes because of the particular gene. So these are some kind of gaps still has to be addressed, though we have knockout technology and overexpression technologies are available, which are all still simplistic. So one more thing, there is a wider gap between a chemical and biological space. So in case of, chemical space, there are more than 10 to the power of 20. The biological space is more than 10 to the power of 4 to 2015. So this is the space, we don't know how, still we don't know that how many proteins or how many target can be available in a particular microbes. You can imagine that even the microbe itself invisible. Inside the microbes, we have the proteins and targets, and we don't know how many millions or how many thousands are available. So obviously, as of now, we are trying to use the computational tools like, molecular docking and screening, visual screening, to try to match that chemical library to the biological space. But still, even though you try to say that it is everywhere, because any molecule will bind with any surface of the protein. But once again, nothing but how far you're going to validate it in the in vivo system, it is called as the quite questionable challenges even now where we are changing it. And really, it is one of the still the unrevealed, beyond the prediction, beyond the computational. Even though we have molecular docking, you can able to say, you say that my tool is showing that is a more potent, it's a promising, best post compound, all those terminology that we are speaking out. But still, the integrated response of the chemistry and biology has to be addressed. For example, and you can see, you're aware of that metronidazole drug, which is more commonly used for amoebiasis, also for the anaerobic bacteria. You can see that there is a chemistry here, and there is a primary alcohol group, which has a nitrogen 1 position. That is the MIC EC50, activity concentration is 2630 nanomole. Just there is a small change where we are trying to modify that particular OH group into phenylethylene group. So there is a relatively significant increase in the lipophilicity of the metronidazole. That EC50 value has come down to 39 nanomole. I don't ensure that molecule is non-toxic, but still The molecular potency has come down a small modification of Vogel's group into the phenylethylamine moiety. It has reduced like anything. So this kind of analogy, this kind of library, and it is completely, even what is predicted in the molecular docking and the binding or posing, it is totally different from the practical reality. The another is, this is, I don't require to discuss much more, the tetracycline. Epimerization and tetracycline. And nobody knows now still how many antibiotics can be epimerized by antibiotic. Whether the antibiotics are inactive because in drug resistance, it's because of other mechanism or kind of epimerization like tetracycline that has to be revealed for all the organisms. And that's what we are lacking knowledge, that what exactly that how the chemistry and biological chemistry of this particular microbes are working out inside the cell. That is maybe the reason for the drug tolerance. And this is, this is the source of nature, that for various models of antibiotic drug discovery and development. There are 4 different era of drug discovery. One is golden era. The second, in around 1975, it was a medicinal chemistry era. In 2000, it was a resistant era. That's what when the drug resistance started, and it was called as a resistant era. In resistant era, we used various tools like computational tools, modern drug discovery, target based. We tried to discover broad spectrum antibiotic, and it was low success. And but when we come for the narrow spectrum era, that's what I'm saying now. Every microbes, once it developed resistance, they are unique. So there is a need for determining the particular drug for a particular resistant species. For example, now the question is here in tuberculosis. Sometimes the regimen is working out for MDR-TB, but it is not working out with the XDR-TB. The reason nothing but the genetic profile or genetic expression of XDR-TB is totally different or significantly different from MDR-TB. Similarly, now we have that, only Pseudomonas aeruginosa. We have MDR Pseudomonas aeruginosa. It is where the aminoglycoside not at all working at all. But there are some cases that drug-resistant tuberculosis, like gentamicin-resistant Pseudomonas. In gentamicin-resistant Pseudomonas, the other aminoglycosides are working, but the same gentamicin-resistant Pseudomonas, it's not responding to other antibiotics. So now that's why you're thinking, but when the organism evolved resistance, whether it is a multi-drug resistance or extensively drug resistance or single antibiotic resistance, so the genome profiles are differing, where we need to integrate and we need to go for a narrow spectrum era, and where we have to integrate unconventional drug discovery also here. Rather going for modern drug discovery, there is a need for unconventional drug discovery, but the success also, it is very predicted. So coming to the opportunity in the plant and herbal based research, and I would like to insist you all to have this, medicinal plant genomic research, and which will help you out to go for, finding out your genome, which is responsible for the particular phytochemical constituents. For example, you can say that in the screen, you can see that there are 2 plants. One is Tulsi, you are aware of that, Osimum species. Another is the Salvia species. They are from different family, but still both the plant has the same gene or the genome we called as copallyl diphosphate synthesis, which is responsible for producing the same type of terpenoids. So here both the plants are effective, and both the plants are affecting the same disease. Similarly, in case of your bitter gourd, the bitter gourd, there is a polypeptide P or P insulin type peptide, which is responsible for hypoglycemia effect of diabetics. Similar way, where you can take your all the plant which are active, which are producing your potent antimicrobial agent, You can try to trace out the particular genome of the plant which is responsible for the particular class of phytochemical constituent. Then you can integrate with the other plant, can able to identify the other thing. So next thing, but I think you are aware of that. Along with the vaccines, the herbal vaccines are the rise. The herbal vaccine terminology is not very new. It is introduced and coined and is mentioned by WHO in 2005 itself. So people are believing that the adverse effect of vaccine from plants are relatively very less when compared to the vaccine from the living beings, like microbial vaccines. So there are many reports I can show you here. Like that, in 1997 there was a plant-based vaccines. In 2009 also there were a report on plant-produced vaccines, and there is a vaccine. There is a mushroom. It's from Japan variety. They developed a vaccine for hepatitis B. It's oral vaccine. They developed this particular mushroom, transgenic Flammulina veletipis. And now in case of synthetic chemistry, it's a growing interest nowadays, especially in case of potentiation approach in cancer as well as antimicrobial resistance. It is just one simple example where you can say that there are phenothiazine. It is a well-known anti-cancer agent. You're aware of that. And the down molecule, it is kind of piperazine hybrid with furanone mighty. It is also a well-known, reported anti-tumor agent. Where you're trying to hybrid these two molecules, you can get a molecule. This molecule is almost more potent than 5-fluorouracil. So now, similarly, in its same approaches, now it is evolving in the antimicrobial research. The people are trying to hybrid two different molecules which are acting on different mechanisms or same mechanisms, and such a way that they're trying to potentiate the molecule. But once again, this strategy, it is very essential. Here we consider phenothiazine, and one thing is that we are to consider the toxicity of the particular core molecule. So now the challenge in drug discovery, not only to get a potent molecule, it is also to see that the residual potent molecules are how far toxic. That's what I'm saying. In the clinical trials, many of the molecules are proven to be efficacious. I mean, not efficacious, proven to be a potent. It may be a, hit to lead optimization is a quite difficult task here. Minimize the toxicity. No, a best example I can call the beta-choline, that beta-choline, linezolid, and these are the molecules entered in ardent drug in tuberculosis. But still now there is a controversy, not controversy, it's absolute evidence that, and the beta-choline is a cardiotoxic now. So that now, and GSK or some companies they're trying to modify this beta-choline, and they're trying to derive this beta-choline, which are non-cardiotoxic agents. So that's all things are happening, which you are supposed to be aware of it. And it is also aware that in the recent time, there are the multi-targeted drugs are so growing interest. And it is also not new. In 5 years ago, in 2017 itself, almost about 30, almost about, almost about around how much percent? 21%, yes. Almost about 21% of the drugs approved by FDA, which are of, a kind of multi-drug targeted drugs only. And once again, if at all, if you want to go into multi-drug targeted drugs, and you need to integrate all the CAD tool, QSAR, and molecular docking, pharmacophore models, and de novo synthesis, etc., etc. It's very essential. And this is a well-aware concept that repurposing drug. You are aware of that. The repurposing is one of the immediate solution which you have come across in COVID also. Once the COVID pandemic is started, and all industries started screening all the drug molecule against the COVID virus. They're trying to repurpose it. So always, to say that repurposing strategy is the immediate strategy for any disease drug treatment. It's very important strategy, and it can be done in very narrow span. And you are aware of the best example, metformin. It's a diabetic drug, but still it is effective in cancer, and it's effective in tuberculosis, effective in memory loss, effective in weight loss. I don't know whether you're aware of that metformin now it's in the clinical trial for tuberculosis also. Similarly, there is a concept as a repositioning strategy. So then come for the repositioning strategy, it's very important thing, but already a drug is discovered and with a known mechanism and known target. But now we are trying to, same molecule is, we are expecting this kind of multi-target only, where it is binding with the other target that giving a different indication, different use. You are aware of that thalidomide is every pharmacy student aware of this molecule, which thalidomide tragedy we studied. So actually it has the property of TNF alpha inhibitions, TNF alpha inhibitions. Okay, so that's why here, and it's kind of repositioning, where one molecule known mechanism is taken into other mechanisms. Okay, and this is also one simple example I want to showcase to you, the thing, but it's a kind of complementary biological effect. I don't know whether you might have come across or not, and this is in between your anti-Alzheimer's disease and diabetics. Most of the anti-diabetic molecules like insulin, amylin, DPP4 inhibitor, GLPR, PPR gamma, metformin, all these drugs has significant or considerable effect in Alzheimer's disease. Similarly, it is also expected that the molecule which has the anti-Alzheimer's potential will have considerable effect in case of your, excuse me, yeah. So the similarly in reverse, and this anti-Alzheimer's agent which are literature that can be taken into consideration, you can consider. So similarly, what happened, you can also put it in case of antibiotic drug discovery also. You can see that complementary biological effect of one therapeutic agent into another therapeutic agent. And it is a kind of your new output or new literature. In recent time, you can see that LSRT kinase inhibitors for antibiotic. And it was really because in almost in 2020, and it's a kind of inhibitor which is acting on the quorum sensing cascade of bacteria, which were trying to use to block the biofilm formations. But the reason, nothing but over the period of time, it is so, though it shows potency, even you can see that the potency of these molecules are 0.6 micromolar concentration. But still, over the period, the organism is, once if you use the LSRK inhibitor, the organism start using the alternative pathway in the TPD. Okay, that is nothing but it's a kind of synthesis, the 4,5-dihydroxy pentane, pentane-dione synthesis. It's a kind of metabolite which is essential in the microbial mechanisms, microbial survival mechanisms. So obviously, what happened? This attempt become what they called as the failure. So why I'm telling you thing, but it is a way that most of the antimicrobial drug discovery and the new drug targets are getting failed. So the LSRK kinase inhibitors failed because microbes can take alternative pathway to synthesize a TPD. So this is a reason that why I told in the first slide, I think, but de-risking at the early stage very important. You are supposed to validate your particular target, whether it's appropriate or not. If you target a one target and you have to ensure that particular microbe should not choose the metabolic pathway or should not have other alternate target, it is very essential, but it's very challenging now. It's very difficult. We need integration of different disciplines. That is what I'm saying, that you are supposed to correlate or try to incorporate gene expression profiling of bacteria like metabolomics and proteomics, and which can help you out and for de-risking the various fact and risk in the early stage of drug discovery. So I'm almost like ending my sessions, and there is one concept is anti-evolution track. And this anti-evolution track, nothing but it's a kind of it's kind of which can try to minimize the evolution of the particular microbes to the antibiotic. So it is the concept that when you give only antibiotics, already the microbes has the phenomena of developing resistance and showing resistance to drugs. So here what you can do, that you can combine your antibiotic with anti-evolution drugs, so that what happen, you will try to minimize the mutation rate, where you can try to minimize the resistant response of the microbes, so that you may enhance the efficacy of antibiotic. It is only hypothesis, it is introduced, and now still the drug discovery are still the attempts are in progress. And this is also a chemistry attempt, like a supramolecular chemistry for drug resistance. And they are trying to incorporate antibiotic in the macromolecule system, and they're trying to use for drug resistance, but ultimately it is failed, and there is no acceptable efficacy was found with the drug resistance, because of the high toxicity, less stability, and low permeability, because it's called a supramolecular macromolecular system, where which cannot, even though you are going to load the antibiotic in the macromolecular system, it cannot cross the cell membrane, so that ultimately it is failed. And this is for your knowledge that new targets and molecular investigations, even now as of today, the protein synthesis are targeted. The one of the target is a peptide deformylase. It is one of the target in the clinical trial people are trying to investigate. And also now more commonly, they're trying to target all the neuropeptidase, like, which are involved in the cell wall synthesis. Neuropeptidase A, B, C, D, E, F, G, Y. So, like, there are different neuropeptidase or A enzymes are available, but obviously this is also quite questionable, where we are not able to target. If at all, if you are going to target this neuropeptidase, and we require multi-targeting, purely targeting neuro A or purely targeting B or C, it is very difficult to expect the bactericidal effect. It until unless you design a molecule that way, I think, but the particular molecule can target the neuropeptides synthesis at different site, like A, B, C. Then it is the synergistically it is possible. So with this, I thank the organizers and the principal, faculty, and the organizers, students of Santirama College of Pharmacy. And also my gratitude to the AICTE New Delhi for the provided fund and for inviting me to this particular 2 days international conference. And so I hope I've done my job to the team. And with this, I conclude my session. Thank you, and all. Over to the organizer.
Sir, thank you very much, sir. Now we request any of the participants to have your panel discussion, interaction with the key speaker. We are expecting any question here from the participants.
Good morning, everyone. So it's my pleasure to thank Ramalingam sir for kindly accepting the invitation on this occasion. And sir has enlightened us about the drug resistance. Most of the researchers concentrate on the anti-cancer and related drugs, but sir has given us that in future the microbes can face a threat to the humans. So rather than the cancer and related, I think we should focus also on the microbes which are a threat in the near future, because drug resistance is one phenomenon which is going to shell out the money of the patients. So thank you once again, sir. Thank you very much.
Thank you. And Sir is there? Mausam cities are there?
Yeah, yes, sir. Sir is there, sir.
Sir. Video. Okay.
Sir, thank you very much, sir, for your excellent talk.
Okay.
Sir, on behalf of Shantiram College of
Thank you, Ram. How are you?
Sir, how are you, sir?
Yeah,
Thank you, sir. Thank you very much.
yeah, need your kind support forever.
Sure, sir. Sure, definitely. And sorry for the interruption. Because initially, when at the beginning they immediately called for some official work, I went and came back, and all things are there. I think I have joined the right time. I have joined the right time, I think.
Thank you, sir.
Okay,
Nice
sir.
to
Thank you, sir. Thank you. Bye.
Thank you.
Sir, thank you very much, sir. Sir, on behalf of Shantiram College of Pharmacy, we kindly request you, sir, to kindly accept our virtual felicitation. Sir, thank you very much, sir.
Thank you. Thank you. Thank you, Munir and the organizing committee. Thank you very much. And with your permission, I am leaving it. Thank you.
Sir, now I take a privilege to invite our profound speaker, Dr. C. Rajareddy Garu, Senior Scientist, Department of Organic Chemistry, CSIR IICT, Hyderabad, Telangana. I would like to give a brief note about Rajareddy Garu. Dr. C. Rajareddy Garu Dr. C. Rajareddy Garu has obtained BSc from Kakatiya University in 1994 and MSc from Usmania University in 1997. After completion of PhD at CSIR, Indian Institute of Chemical Technology in 2002, Sir has moved as a postdoctoral fellow to University of South Florida, Tampa, USA, and subsequently to University of Mississippi, USA, 2002 to 2005. He returned to India in 2005 and joined as a principal scientist in Sai Life Sciences, Hyderabad. After 1 year, he joined CSIR IICT, Hyderabad as a scientist at Department of Organic Synthesis and Process Chemistry, and presently working as a senior principal scientist. His research interests are both fundamental and applied research, which includes the chemistry of propagylic synthesis, synthesis of polyaromatic hydrocarbons, and bioactive natural alkaloids, process development of APIs such as favipiravir, remdesivir, pregabalin, key fragment of erbilin mesylate and TLR 7/8 agonist molecule used as adjuvant in COVID-19 vaccine have been developed and transferred to various organizations. Sir is a recipient of CSIR Technology Award 2021, NASI Reliance Industries Platinum Jubilee Award in the year 2020, and CSIR Technology Award simultaneously in the year 2020, CRSI Bronze Medal in the year 2018, CDRI Drug Research Excellence Award in the year 2017, and Dr. A.K. Singh Memorial Young Scientist Award in the year 2014, and Evra Young Scientist Award in 2011, and AP Academy Young Scientist Award in the year 2007. Sir is a fellow of Telangana Academy of Sciences in the year 2019. And to his credentials, Sir is an author of 143 publications, 10 patents, 3 review articles, and 2 book chapters. Under his supervision, 23 students have been awarded PhD degree. Presently, 12 research fellows are working for their PhD. He has also supervised more than 20 master students for their dissertation. Now, I kindly request Dr. C.H. Rajareddy Garu to kindly deliver your talk. Sir, please. Sir, we request you, sir, for kindly share the presentation.
Yeah, thank you. Thank you for your kind introduction. So, first of all, I would like to thank the organizing committee of this conference, in particularly Dr. Shivashankar Reddy, and for kind invitation, giving me opportunity to share some of my views and experiences towards drug discovery program and what we are doing at IICT in this direction. So it's a great opportunity. So now just give a moment so that I will share my screen. So hope you are able to see, right?
Sir, we can see.
Is it in presentation mode?
Sir, no, sir. Presentation is visible, sir.
Okay. Okay. Thank you. Thank you for the confirmation. So what I thought of sharing as an organic chemist, what is the role, how it plays in drug discovery, and some of the experiences in the last couple of years under the pandemic conditions, how we contributed for the COVID-19 mitigation, and in a broad perspective, because the audience and others are from mostly pharmacy background and some of the research scholars are there. Overall, how chemistry plays a key role in our day-to-day life. So this is what I thought of sharing. So in this presentation, I would like to show these or cover these kind of topics. Initially, role of chemistry in life, and mainly drug discovery. And in drug discovery, this organic synthesis, organic chemistry is one of the important role it plays, and what is our experience, and slowly to the what is drug repurposing. This especially we have used this during COVID-19 pandemic, and then some of our contributions. So wherever I go, I show this as a first slide to show the importance of chemistry in life. Most of us, when we, especially school children, or who doesn't know much about chemistry, as soon as you say chemical means something, it is a kind of poison chemical, so we should not touch, and like that kind of feeling everybody will get into their minds. But if you really see, everything is a chemical. Even for that matter, what we drink water, it's again a chemical. It has a formula H2O. So we need to have that kind of perception about the chemical, and without this, there is no life. Right? Without water, we cannot live, and all our body is full of chemicals. And in our life, morning when we get up, we start with our day with brushing our teeth. Again, whether you take a brush or you take a paste, everything is a chemical. Until you go to bed in the night, again you have to use your all out or any other mosquito repellent. And in between, from morning to evening, you deal with so many chemicals. Your soap is chemical, your paint is chemical, your shirt is having chemical. So like that, you imagine anything, you cannot imagine any item or any thing without a chemical. You cannot imagine. So this is how it is everywhere. Chemistry is everywhere. It is there in food, it is there in bad. It is there in explosives, it is there in flavors, it is there in our body deodorants, it is there everywhere. You take, whether it can be agrochemical, it may be petrochemical, it may be pharmaceutical, anything. It may be any material, your mobiles. In your mobile, all the materials are made up of with some kind of chemical. So that is where I think it is very important for all of us to understand this, how it is playing a key role in our life. So even during the last couple of years, during the pandemic situation, again everything, chemistry played a key role. Whether you take a testing kits, kind of reagents, standards, RNA extraction, or you take hand wash solutions, sanitizers, or for treatment pharmaceuticals, and more than 100 vaccine projects are going on. Some of the vaccines are into the market. Again, everything with a chemical. So coming to the topic of the seminar, which is a very important topic, and Dr. Ramalingam gave some brief outline. What is, drug discovery, how it is going to, and where we should focus. In addition to that, what I wanted to show here, how our drug discovery era started thousands or hundreds of years ago. It all started from nature. Nature is the inspiration for our drug discovery. So olden days, the treatments are in very crude way. That's called, natural products we used as a medicine. Throughout the history, it is there. Till today, I think in most of our villages and other places, these traditional medicines are being used. But what this medicine contains? It contains a molecule, a bioactive molecule. You all know that, you name any plant, we know that what kind of chemicals these plants are having, whether it may be leaf, it may be root, it may be stem. So this is where our drug discovery started. And the best example, aspirin. How, aspirin got discovered, all of you know, it is a plant material, and then a small modification. Salicylic acid is a plant material, and when salicylic acid got acetylated, acetyl salicylic acid became a aspirin. So the inspiration is from nature. So if you take nature and our natural products, particularly for drug discovery, we can divide into 4 different parts based on their origin. If the natural product is coming from microbial sources, it's a microbial world. If it is a plant source which is growing on our earth, it's a plant source. The best example is paclitaxel, which is used for cancer treatment, breast cancer treatment. Basically, it comes from Texas brevifolia plant. And if the natural product is from marine sources. It's called marine natural product. Best examples, again, anti-cancer compound, discodermadiol and other. And if it is animal source, best example is epibetidine. It is isolated from African cloud frog. So this is how olden days, whether it is Chinese or Indians or North Africans, there is a lot of evidence that using of these natural products for curing various diseases. So, such a beautiful nature providing these chemicals. Why? The nature is providing or producing chemicals for their survivals. They need to survive to these conditions. So accordingly, they produce these chemicals. Now we are taking advantage of these chemicals as a source of inspiration for our drug discovery. So few examples I will show here. Morphine. It is one of the oldest drug known for mankind, and it is used for various treatments. Asthma, sometimes bad eyesight, a bit diarrhea, like this. But originally, this alkaloid is isolated from opium. That's how it got named morphine. So similarly, there are several other natural products became drugs into the market. Here I am showing some of these examples. Don't worry too much about the structure of the compound, but these are all natural products. You can see here, see this is the salicin, is the natural product. From this, we made acetyl salicylic acid. This is called our aspirin. This is morphine, just now I explained, and you can see compound number 4 is digitoxin, is again a natural product. Quinine we use as an anti-malarial compound number 5. This is the quinine structure. This is again a natural product. So similarly, pilocarpine is a imidazole based compound. It is again approved by FDA for dry mouth treatment. So like this, various natural products either directly or indirectly in the form of their derivatives have been approved by FDA for the treatment of various diseases. So this is a history. We are going through history, then we will come to the present situation. So these are all, you can see from the structure, antibiotic compounds, lactams. All these antibiotics are natural products, most. Penicillin, your narcolepsin, your imipenem, or your azithromycin, you name any of these antibiotics, these are all isolated from nature. That's how we could able to discover these antibiotics. This slide shows anti-cancer compounds. Doxorubicin, it is used to treat acute leukemia and various other cancers. It is isolated from Streptomyces fasciitis. Similarly, you look at paclitaxel. It is again derivative of taxa. Taxus brevifolia. This is the tree. You can see a big tree. From there, we isolate this compound baccatin 3 and you convert into a different derivative by attaching the side chain. It became a drug. So from this, we can clearly see that nature is our big inspiration for our drug discovery. Whatever we are doing, now we are modifying these structures to improve their efficacy, or to improve their other parameters to become an efficient drug. These are some of the recent examples where again natural products approved as a drugs. Artemisinin is again isolated from the plant Artemisia annua. It is an antimalarial drug approved. So here, this is an interesting story. Halicandrin B is a marine natural product. It is isolated from different sponges, mainly called Halicandria vocadoi. So while you all noticed that majority of our drugs mostly from natural sources, but one thing we have to notice that, see, nature is producing this chemical, but when we want to use for our mankind or for the required population, do you think the quantity of this material produced by nature is going to sufficient? No. Even we cannot do the initial biological studies and testings before even getting approved market, the quantity is not sufficient. Just you take the example of this Halicandrin B. When it was isolated, it was only few milligrams, maybe hardly 5 to 10 milligrams. From that 10 milligrams, natural product scientists confirmed its structure by various spectroscopic techniques. So this is the Halicandrin structure. But now, do you think that this 5 to 10 milligrams is going to enough to study all the studies? No. You need to do lot of toxicology studies, pharmacokinetic studies, then you go to phase 1, phase 2, phase 3. Before it getting to approval, it has to undergo several safety, efficacy, stability, several studies has to be done. These all required kilograms of compound. And even if it is approved, when you want to come into the market, you need to have enough quantities of this compound. Sometimes maybe some 100 kilograms, maybe ton scale. You imagine how much paracetamol whole world is consuming. You just think your paracetamol is 600 milligram tablet or 500 milligram tablet. So per day, if you take 3 tablets, it is you are consuming 1.5 to 1.8 grams of per person. And you imagine all over the world how many people will take everyday paracetamol, and then how many tons of paracetamol you require. From where it is coming? So this is where organic synthesis plays a key role to synthesize this compound in required quantities from the available resources. That's the art of science called organic synthesis. So coming back to this slide, this halicandrin, they isolated 5 to 10 milligrams, and there is no enough sufficient quantity of compound to study further biology. So then in Harvard University, Professor Kishi group has started synthesis of this compound. So during their synthesis, you can see that this right side portion of the compound, when they completed synthesis, half this portion, for curiosity, they wanted to test. Then see, this is showed much better activity than your original natural product. That's how this half structure of this halicandrin B became a drug in the market now, ergoline mesylate. It is called, and this is the recent drug used for the treatment of breast cancer. Because of its complexity, the synthesis of this compound is very challenging, and it's very expensive molecule. Per injection, $2,000 to $4,000, and you need at least 5 injections per course. So we at IICT made a new process for this molecule, and we transferred to a couple of companies. They are now evaluating to bring it to the market for cheaper price. So this is one of the activity we do at CSR IICT as a major, of course, additional to other activities. So coming back to this lecture, so now, as I mentioned, nature is our inspiration for drug discovery, and from nature we are learning the lessons. But without destroying the nature, we should take that lead and we should move further. Because if you want to have tons of compound of this molecule, where do you isolate? You cannot collect that much sponge from the sea. And that collection of sea in the sponge also, it is very particular time to time. Because that sponge in April, it may produce a different chemical. In May, it produces a different chemical. After 6 months, it produces a different chemical. So there are a lot of things matters. That is where the drug discovery process involves a lot of steps. So in reality, it requires so many stages. Initially, we do in vitro studies. Then when you know our compound is active, we go for in vivo validation. Then we have to screen lot of compounds and identify lead. Then after studying all the pharmacokinetic studies, bioavailability, safety, toxicology, then we filter from 5,000 compounds to 5 compounds. This is only a rough idea. It is not every time 5,000 compounds. Sometimes it may be 50 compounds to bring down to one molecule. We never know. But this is a general process I am showing here. So once you have the lead compound with safety, toxicology, PK, PD, all studies done, then it goes for clinical development. Again, where phase 1, phase 2, phase 3. Initially on small animals, then big animals, and then go to human clinical trials. And finally, one compound may come into the market or it may fail at any stage. So that is where drug development process is very expensive. And on an average, this is the expectation of $2 billion, and it takes 10 to 15 years. So it requires a lot of branches. You need pharmacology expertise, you need toxicology expertise, you need synthetic expertise, you need so many things. You need analytical experience. So it is not a one person or one branch can develop a drug. It's a lot of branches of sciences put together involved in the development of drug discovery. So as I mentioned, the synthesis plays important role. That is where our expertise at IICT, our research group is mainly working on synthesis. So why we need to synthesize these molecules? I told you already one reason, that when natural products showed or inspired us with its structure, we have to look at the structure and plan our synthetic strategy. How can we make this molecule in the lab? When nature is making itself, that means it is possible to make in the lab also. So that is how we plan. That is one of the reasons. But at the same time, sometimes that structure, because that structure of molecule is being created or produced by the plant for its own protection. But the same compound may not be useful to the, as such, as it is, it may not be useful to our treatment. So sometimes we can do some modifications. So new scaffolds are expected from our academic institutions, so that these scaffolds are inspired by the nature, which can go for further evaluations. So synthesis is a discipline. It is a central for our areas of chemistry. So the chemical synthesis means it is an intentional construction of a chemical molecule, usually natural product. So from commercially available starting materials, maybe glucose, maybe some amino acids, maybe some of the other natural products are largely available in large quantities, which we need to plan and convert into the required molecule. So that is called a total synthesis. If we are making a natural product, it is called natural product total synthesis. So why we have to make these natural products in lab? There are three reasons in my perspective. The first one is, once we know the structure of the molecule produced by the nature, it really posing challenge to the mankind by developing the new reactions or by using the existing chemical reactions, whether can we make this molecule in the lab. So because it is a complex structure, it inspires the chemist to develop new reactions. Similarly, this point I have already mentioned, that total synthesis is essential because we need to make in more quantities for further biological evolution. Other option, we can make some of the analogs. Sometimes, because of it is very low quantities available, sometimes the structure may be wrongly given by natural product, because end of the day, he is another scientist, is a human being. We will do some mistakes in some times. So if you see the best example, quinine, this was the cholesterol. This cholesterol structure is proposed like this initially, but cholesterol structure later it is modified, because when it got isolated, it is showed like this. But after synthesis, it is not matching, then we have to revise. Then natural product synthesis provides important forum, and we can develop new methods and we can show these things. So this is the reason, 4 reasons we can show clearly why we have to make natural product, because we cannot destroy the nature. If you want ton quantities, we cannot cut all the trees and extract what you want. Then how do you grow that? So this is where synthesis plays a key role. Now I will give you a few examples, importance of the synthesis history. All of you know that urea, it is again natural product, first discovered in 1727 from urine. You take the example of urea, how many tons we required? All farmers home, you can see these bags of urea in their houses or in their fields. We have to use for protection of our crops. So we required in ton scale, we have to produce. But can you imagine, is it possible to isolate the ton scale from urine? No. You can see after 100 years, it is exactly 100 years. It was isolated in 1727. In 1828, Frederick Kohler developed a process to synthesize this compound in the lab using ammonium chloride and silver isocyanate. These are plenty available chemicals. So from this, he made a process for synthesis of urea. That's how today we are able to use this urea for our crop protection. So this is the first molecule made in the laboratory, organic molecule made in the laboratory in 1828. Very simple molecule, 1 carbon, 1 oxygen, 2 nitrogen, 4 hydrogen. But now close to more than 200 years, or close to 300 years now from 1828, how chemistry developed. Today, we can do such a complex molecule, not only the simple urea. You see some of the examples. Camphor, Again, it is a natural product. It was isolated in 1903. Before that, but 1903, first synthesis of this discovered from pinene. How much camphor we consume every day in lot of temples and everywhere we required in large quantities. If you want to extract from this our trees, how many trees you have to cut every day? I think our forests also not sufficient if you want to do this. So again, you have to make in the lab. Similarly, quinine is an anti-malarial compound, again isolated from nature. But this is where the modern era started for total synthesis of a molecule, especially Woodward's, a Nobel laureate scientist from US. He made this compound first time in the lab. You can see it has a chiral center. Now this one molecule can be in R isomer or maybe S isomer, but only one isomer is active. So you have to selectively make that isomer. So that is the challenge and art of synthesis. Coming to 20th century, see Professor E.J. Corey, Harvard University. Again, he is a winner of Nobel Prize in 1990 for his contributions in organic synthesis. Because no spectroscopic methods were available those days, but still he could make such a important complex molecules which mostly are being used as a drugs today. Prostaglandin we use in various treatment as steroids in our treatments. So having said, from urea to complex molecules, today biggest challenge the scientists are trying to make this molecule. Can you look at the structure? It's mycotoxin. It's very, very potent compound. We cannot handle. You see, alphabets also not sufficient to give the nomenclature. Again, we have to give after A to Z. You have to start with A dash, B dash, C dash like that. So, but if you look at other way, the technologies, the spectroscopic methods, the analytical methods are so advanced today, a man can make any kind of complex molecule. It's a matter of time. So this is about synthesis, and I think so far we have learned how chemistry plays an important role in our life and how our drug discovery era started from natural products and how we are using that knowledge intelligently to convert into our healthcare treatment pharmaceuticals, and how organic synthesis is playing a key role to make these molecules, and what is the trend from the day urea has been made in 1828 until today in a 300 years, how our chemistry progressed. So now I would like to show some of the activities in our research lab at AICTE, and then how it is very important, because synthetic making a molecule in the lab is easy. Maybe in lab you can make 1 gram, 5 gram, 10 gram, maybe 1 kg. But when you need in ton scale, you need to build big plants. And again, there engineering plays a key role, chemical engineering. So there is several aspects are involved. It's not just like that one lab or one organization can make a drug and produce. It involves a lot of steps. So what we do at IICT, I'm not going into chemistry, reactions, mechanisms. So many things are involved, but I'm not going into that direction. I am trying to give a overall idea of chemistry, how it plays a key role in drug discovery. So you have seen that how we got inspired for our drug discovery. Fine. Now, how we are using our knowledge to make new molecules? Because now we have explored a lot from the nature. By inspiring, we have developed a lot of pharmaceuticals and drugs. But every day, new diseases are coming. New targets have been discovered, and which requires new molecules. Again, you need to use our chemistry knowledge to make new molecules. Sometimes a molecule not in the nature, but we can make in the lab that may be useful for further drug discovery programs. So we do 3 different activities in our lab. This is one of the activity, bioactive natural products, which I have shown already. We will take the nature as inspiration for chemical structure, then we try to do in the lab. Sometimes it takes 5 years, sometimes it takes 5 months, sometimes we fail. Most of the PhD students and other project assistants will work on that. Again, within the natural products, my specialization is macrolides, alkaloids, and these kind of compounds. To do these things, we need to develop new methodologies. So our main focus is cyclization reactions of these 2 starting materials, propargylic alcohols and amides, to make different heterocycles and carbocycles. So this is our 50% of research activity we focus. But in addition, our organization name itself says CSIR, Council for Scientific and Industrial Research. We need to address the industry problems, support industries. So that is where we do a lot of industry collaborative research, either to make new chemical entities for drug discovery, or we develop a process to make the compound or a drug to become much cheaper or a environmentally friendly process, so that we can stop polluting our country or by producing these compounds. So don't worry about these structures. Just I have shown some structures. So these are the natural products we made in our laboratory in the last 15 years, working almost. In 2008, the first molecule we made called centralobin. It is a tetrahydropyran molecule isolated from nature. Again, we made ryoeptilol. We developed a route for this diaspongin. Like that, you can see slowly we improved increased the complexity. When I started in 2008 my career, we have taken very simple molecule having 2 asymmetric centers. But slowly we have taken complex molecules and we are able to successfully make these compounds in gram quantities. These are some of the other macrolide natural products we again made in our lab. This particular C-metopolide compound, when we made, we realized that natural product scientists mistakenly proposed its stereochemistry, but through our synthesis we have corrected. Because he has a small amount of compound, he could not characterize properly the absolute configuration. But since we made, now our data is not matching with that the reported data, then we have to make all the possible analogs and then we have to revise the stereochemistry. Similarly, alkaloid molecules, presently we are concentrating. So are you able to hear me? Hello. Hello.
So sorry for net connectivity, sir.
Are you able to hear me now?
Yes, sir. Audible,
Okay.
sir.
Okay. Okay. Thank you. So these are some of the alkalides which we have made in our lab, and presently we are addressing this complex molecule synthesis. So in the process, all these PhD students and everybody will get a training of how to make a molecule, how to plan, what strategy we have to use. So I will show one example how we plan in the lab to make a molecule. You see, this is a cladospolite D molecule. It was isolated in 2001, but when it was isolated, only they got 5 milligrams of this compound from this fungus on the corn. When this kind of infected corn they will take, and they will put in some solvent, then they extract the compound from that solvent, then they decided the structure is like this. So this structure, but if you can see that this red color part is very challenging to make this cis double bond, and that is where the challenge in this molecule. So we have taken up this challenge in some time 2010, and we used this approach. These chemicals are available in the lab. I'm not going to explain any chemistry, but this is how it involves when you wanted to do a molecule synthesis in lab. We have started from this kilogram scale available epoxide, and slowly we built all the required carbons, oxygens, and everything. Finally, we prepared a 500 milligrams of this compound. So this is a just example of how we do in the lab different reactions. Some of you learned some named reactions: hydrolysis, protection, dihydroxylation, oxidation, reduction. So all these things we have to use in the lab. Similarly, this is another, longan lactone, isolated from longan seeds in China, and it has been used. This fruit they used to for the treatment of various compounds. But since they isolated only 3 milligrams from 10.5 kilograms of seeds, and there is no biological activity because it is only few milligrams, then we took this. It got inspired our chemists. Some student came up with this compound. Sir, this compound is very interesting. Till now, nobody has made. Maybe it is interesting to study biological activities. Yes, please go ahead. So then the students started, within 6 months of period, from available L-aspartic acid, by doing around 8 reactions, we could make this compound in 200 milligram scale from 375 milligrams of L-aspartic acid in just 6 steps. So this is the power of synthesis, and we can make both the isomers. Natural product having only one isomer, we can make both the isomers. Which isomer may be active, we don't know unless we test. So now we are doing biological testing of that particular molecule. So this, don't worry about this, just I am showing that how we develop a new methods in lab. So many new methods we develop for synthesis of heterocycles and carbocycles, and all this will publish in different journals, international journals. Similarly, you can see this is another type of approach we are following. A common starting material we start with. By doing different kind of reactions, we can make different heterocycles and carbocycles. Again, all these things we are publishing since last 10 years, from 2011 you can see up to 2021 we are doing this synthesis from this material. So this is a routine approach in our laboratory. What are the recent advances? I wanted to update and show here. One topic in today's scenario is photo redox catalysis. Because we are using so many oxidating reagents, chemicals in our process, instead to avoid that reagents, whether our visible light or some photochemical light also can do this chemical reaction, so that we will not pollute our system with the byproducts. Because if you are using some large quantities of oxidant, you get a byproduct, then where to dispose that? For that purpose, recent techniques are being developed for, one of the technique is photochemistry. So under the light, you can do reactions. Only you take the starting material, expose to light, and it will undergo some chemical transformation. Only you need to identify which nanometers, under which nanometer it is going to, which kind of transformations. You need to analyze it. This is one thing. The second thing is electrifying organic synthesis. So this is electrochemistry. Means you pass the current, use anode and cathode. Again, they are with metals, but you can transfer one chemical to another chemical by electrochemistry. So again, we can avoid polluting with byproducts. Similarly, doing reactions in one part. I have shown in previous slide synthesis one of the molecule in 6 stages. So each stage we have to isolate the molecule and do the next reaction. That require a lot of chemicals and also solvents, and again it produce lot of waste. Can we do the all 6 reactions in one part? That is challenge. Using different catalyst. So this is another area today people are focusing. So I think now you have learned how drug discovery and how natural products played a role, and then how organic synthesis is playing a key role in production of these compounds, everything. And what are the recent techniques for synthesis? So here I am little bit, changing the gear. In drug discovery, in the recent past 2 years, this repurposing became a buzzword, because you can see here that why we need to take up this repurposing drugs. I have already shown in my first slide, in the beginning slides, because of thousands of new diseases are coming, because of lifestyle change, because of our food habits, or whatever you call. End of the day, it is a natural mission. We don't know what is going to happen in our inside the body. So when such kind of new diseases are coming, if you are taking 10 to 15 years to discover a drug and $2 billion of expenditure money, and for this many diseases, how many years you need to, every time, yearly maybe we are getting some 20-50 drugs into the market, but we are having 100 to 200 diseases in new diseases. So this is a continuous process. So now the repurposing, because in the last 200 years or 100 years, we have discovered so many drugs, but every time for a new disease, we are looking for a new drug. Another way of looking is repurposing. Whether any of our old drugs which are currently being used can be used for a new disease. This is called repurposing. We have discovered for some purpose, but today we are repurposing. because it is already in the market. That means you no need to do lot of other studies like toxicology, pharmacology, and you know the synthesis. You no need to develop any route. Everything is available. Only thing you have to study for a new disease. So it requires less money and also it requires very short time. That is what we did for our COVID-19 mitigation, and quickly some of the antiviral drugs have been launched for use as a COVID-19 treatment. So now this is a new technique for studying the drugs that are already approved to treat one disease. Now whether they are safe and effective for other diseases. That's what called drug repurposing. And it's only 2 steps it requires. You need to screen for a particular target and investigate the minimum number of pathways and enter into the clinical trials very limited so that you can launch into the market under emergency or whenever it required. So now I have shown that we have made so many molecules in our lab, so many heterocycles in our lab. But now you have discovered a drug, you have a synthetic route, it is approved, everything is fine. But as I mentioned, we need to produce in large quantities in required amounts. So that is again a specialized branch. It is called process chemistry. So this is a special art. Doing lab scale is different, production is different. While doing production, you have to consider so many things because your byproducts because you are producing pollution. So you have to avoid that. You need to look at cost. It should be very, very effective cost. Otherwise, you cannot be in the market. So several parameters need to be taken. So I will give a little bit touch upon. It requires again so many parameters, process chemistry. See, here we have started our journey, new chemical entity, NCE. That time we may need 1 gram to 100 gram. But after approval, you need a commercial API. It may be kilograms to tons. So in between, to convert this 1 gram process to ton scale process, you have lot of steps involved: synthetic route, starting material availability, route selection, so many steps involved. So this is just pictorial for some of the students and research scholars. See, when we are doing in our lab, this is the setup. First picture you can see Mr. Amul Patil is monitoring the reaction in maybe 100 to 100 gram scale. Then in the next photo, you can see a couple of students are purifying the compound using column chromatography. And after purification, you need to evaporate the solvent to get our compound using rotary evaporator. Here, Mr. Subbarao is doing the job. So this is all, experience in our lab. But this process, if you want to convert to produce in kilogram ton scale, you can see some of the pictures I have just downloaded from the net. How a reaction chemical plant looks like in the industry. And you need to do in this 1000 liter, 2000 liter capacity, sometimes 10,000 liter capacity. So you need to do a lot of safety precautions, everything. So over a period of in last 75 years, our institute contributed for various diseases. Several drugs and technologies have been developed, transferred. Some of those things won technology award in 2014. You can see this misoprostol. It is used, in abortions and before, to stop the like that 100 microgram. This is a called. Then you knew AZT in 1980s when AIDS was, in peak stage, and this is the only, it is not a real treatment, but it can expand the life of, patient. So it was initially US government approved, but it was so expensive. Then IICT made a contribution and transferred to CIPLA, and CIPLA used to produce and gave in Africa $1 per tablet. So in 2019, you can see that India, 2009, so many people were infected. But today, because of these HIV drugs, now we are under control for this. So very few recent experiences of our COVID-19 during the last 2 years pandemic, what we did. See, when this pandemic was started in 2020, actually 2019 December we know, but in our country it came little late in February, March. We were noticed it is going to be dangerous for our country and whole world. But what can be done? So our institutes all together, we thought of taking a drug repurposing pathway. We identified various potential compounds, around 20 compounds, and we decided that if suppose this compound is approved for the treatment of COVID-19, but how to produce in large quantities. So you can see the right side 12 compounds are available in India. Indian manufacturers are already making these compounds for something. So even if it is approved for COVID-19, we can get these molecules or we can get these medicines. But you see the left side 8 compounds. If they get approved, nobody is there to make in India. So that is where we started developing a process for these 8 molecules, and IICT has taken up first 2 molecules, remdesivir and favipiravir. And I have led personally our team and worked during the pandemic. Everything is closed, but we are inside 4 walls in laboratory. 24/7 we are working, and we developed favipiravir in just 25 days, a new technology. And when it got approved under emergency conditions, in May 2020, number of companies approached and permission for clinical trials. And after few clinical trials, because it has already been used in Japan, like our cetirizine. Favipiravir is being used in Japan as like the way we use for cetirizine. So that is where you don't require so many chemicals, so many testings. But when it was launched by Glenmark with the Japanese technology, the tablet cost was 103 rupees. Then we developed a process and transferred to CIPLA, and they marketed as Ciplenza. You can see here IICT logo in the tablet pack, and that time within a month the price got down for 68 rupees per tablet. But later further studies and many people produced, slowly it became 30 rupees per tablet or 25 rupees per tablet. It's a different story, but later even some people said that it is not even working. So but at the peak stage when nothing was available, this has been used for the treatment of COVID-19. And later, many people know, even a common people know what is remdesivir, how it was hot molecule for COVID-19 injection, remdesivir injection. This is again a new molecule. Only trials were going on. Originally, it was discovered for Ebola virus, but somehow due to some reasons, it could not come into the market. But again, when this coronavirus came, it got tested and trials were started. Numerous companies were, then it was launched under emergency only for patients, COVID-19, severe patients, it will be given in the hospitals. With that condition, it has been approved. But again, IICT played a key role in developing the process and transfer to different companies. So favipiravir we have transferred to 3 companies, CIPLA, Luxi, and Fine Chemicals, and now remdesivir also Mylan, CIPLA, Luxi. And similarly, sometime in June 2020, Bharat Biotech approached us that they want to have one chemical process because they know that that chemical is working as a adjuvant molecule, as an immune booster in the vaccine, but the large production of that chemical is very tough job when it was discovered. So you can see here, it is there in the website. I am not using anything from my own knowledge. So these 6 chemicals are there in the vaccine. You can see Bharat Biotech website if anybody interested in Covaxin, our India's first indigenous vaccine. In these 6 chemicals, you can see one is inactivated virus antigen, other one is aluminum hydroxide, then 2-phenoxy. These are all routinely used chemicals in vaccines. But this particular blue highlighted molecule, TLR7/8 molecule, is new and they found it is very important in this vaccine. But when you want to make this compound, because now you have to produce this vaccine for crore people, not only in India, different countries, and you have to give 2 doses. And this compound, it requires only 15 micrograms. 15 micrograms of this compound will be there in each vaccine. But still you need to produce in kilogram scale. So you see here, when it was initially made in the lab, these kind of reactions are involved. It's very tedious. It is impossible to make 10 grams also. Then we started this project in July 2020 and we made a new process and we gave the first gram of sample in September 2020. Finally, technology transfer in December 2020 with, these are our no purification required. Very simply we can make with Indian available chemicals, and finally we can reduce the cost 10 times reduce the cost. We filed the patent and everything, we transfer technology, and all of you know that in 2021 January, Covaxin got approval for emergency usage. So very timely we developed this technology, and whoever took today Covaxin vaccination in your body, the chemical made by AICTE technology is there in your body. So I am proud to be, team leader of this project, and we also received this technology award for this technology. And we have transferred this to different companies, and they are giving the compound to Bharat. So this is how a science and scientist can play a key role. Not only, I'm not saying that only organic chemistry or organic synthesis, but each branch of science is very important. And we have different laboratories, National Chemical Laboratory, NIST Trivandrum, NIST Jorhat, and the different labs very effectively and productively worked on this COVID-19 different drugs. But finally, some of the compounds into the market, some of them could not pass the clinical trials. And our Director General, Professor Shekhar Mande, our previous director, Shekhar, he being presently the Secretary DST, and our new director, all played a very key role during these 2 years of pandemic time. So this is for all audience, IICT means you may think that we only do chemistry, chemical technology. No, in addition to this, we have various departments and various buildings and various laboratories. During this pandemic time, lot of social services also our institute has been contributed. So through science, we can do everything. Even we can take care of your health, food, social responsibility, agriculture, anything for the quality improvement. We need our science and technology development, which is essential for any country. So you can see our employees are giving sanitizers to municipality and health workers and police, and water. Also, we have water technologies. Water is also supplied, and similarly some food supply distribution at markets, different markets. So this is how we also actively involve towards corporate social responsibilities. So here I would like to conclude my technical talk. Today I am able to be in front of you by giving these results of our laboratory and what we have achieved. This is all because of my very dedicated and energetic, enthusiastic students. I am only mentoring them and choosing the problem, but they are the real workers and heroes in the lab to make it happen. So in the last 15 years, around 22 people have been graduated from my laboratory with PhD, and presently they are working in different parts of the world, and some of them are in academics, some of them are in industry, and presently, you know, around 12 to 13 people are working in my lab for PhD program, apart from various other projects. And our funding agencies, mainly our parent organization CSIR, DST, Ministry of Earth Sciences, of course, SERB, these are all contributing to our basic science. But in the last 3-4 years, a lot of sponsorship came from CIPLA and Bharat Biotech, which needs to be acknowledged. And they gave us opportunity to contribute to COVID-19 mitigation. I'm thankful to all these students as well as funding agencies and all the directors, previous directors and present director, and of course these two industrial organizations. So here I would like to take to you about our institute little bit, because many of you know about this IICT name, but since all of you are away from the institute, and just I wanted to give a glimpse in next 5 minutes about our institute. See, it was actually started, you can see, before our independence, 1944, by Nizam. You can analyze, you can see that now how Nizam King was thought that this kind of lab is required in this region. In 1949, after our independence, it was actually started in Osmania University. But in 1949, this land is allocated by Nizam, and then slowly after independence, the first foundation stone laid down, and 54, Pandit Nehru inaugurated our building. You can see that construction when it was in 1949. And later, for 56, it was taken over by CSIR, and that time they named it as a regional research laboratory. So each region used to have one lab, RRL. Till today, many people know it as RRL, because RRL Hyderabad, RRL Jorhat, RRL Trivandrum, RRL Jammu, like this, each region has a regional research laboratory. But in 1990s or 89, based on the work of the lab, they renamed some of the labs. Now this RRL Hyderabad became Indian Institute of Chemical Technology, because of its technologies mostly developed from chemistry. So it appropriate to give this name. So that's how it has been changed. So these couple of pictures. So there could be no demarcation in scientific progress. That's what Nehru, he told in those days in his speech on 2nd January 1954 when he inaugurated the building. Since then, in 75 years, institute grown up very vastly with lot of centers, center for lipids, center for catalysis, chemical biology, you name any equipment, world-class equipment we have at our institute, and NMR, mask, XRD. It has, polymers, materials, several things we do at IICT, drug discovery research, drug delivery, chemical engineering, design engineering, several departments, natural products lab, even agrochemicals and fluorochemicals, pheromones, like various departments. Whenever some college or somebody wants to visit our institute, you are most welcome to write to us, and so that we will be happy to take up, because we are doing a lot of inspired programs to uh, encourage the students at the school level, college level, to take research as their career, so that the country progress will be in much faster way through science. So these are some of the facilities we have at our institute to analyze our compounds after the synthesis, lot of advanced techniques. So this is the unique facility at CSIR IICT we have. It's called Mole Bank. Here, it is not a money storage bank, but it is a molecule storage bank. Whatever molecules we are making, I have shown, we store in this facility, so that whenever a new target, new disease comes, these molecules can be again screened for new. Today it may not be useful, but tomorrow for a new disease it may be useful. So previously we have thrown our lot of compounds into drainage systems after our PhD is over. So that's how we thought that we are making by using lot of time and energy, money. But after our PhD is done, we make one book and we get our degree, but we are throwing the compound. So to stop that, we have made this facility in 2010. Now this has more than lakh of compounds. Every time we check the purity, and all new targets will be screened. So this is how inside it looks. Everything robotic control, and we takes care of all the purity and time to time. So it goes to further screening in biological laboratory. So this is how it works. Different diseases and the mole bank compounds will be screened using high throughput screening facility at IICT. So finally, just if any B pharmacy, M pharmacy students, this is for everybody, even for research scholars. See, most of the times we work in a comfort zone. We want easy work. We want easy, completion. But always please be remember, research or science development cannot be happen like that. There is no shortcuts for science and development, only hard work. You have to be trained hard and be patient, then only it will pay you at the end. So the choice is now yours. If you want to do easy work, every day you can complain, this is not there, that is not there. You can blame the organization, you can blame the management. So you, whatever, cheating, pretending. So this is easy way of doing. But always a hardworking person, for everything he will get inspired. He try to mentor and he try to learn, he try to help, and he try to giving and taking. So these things, if you develop in your attitude, I think it is very important for a research person to which become a successful scientist. So with this message, I thank you one and all for your kind patience. Here I did not give you a particular topic of drug discovery, but I tried to cover a flavor of drug discovery and how a medicine before coming into the market, what are the steps involved in this, how we got inspired, how our history, and how where we are today, and what we need to do in future. So just I tried to cover a broad perspective because of mixed audience of research scholars and faculty. So I hope you could get some message from these lectures. Again, once again, I thank the organizers for giving me this opportunity to share my thoughts and research experience. With this, I conclude my talk here. Any question, answers, I'm happy to take up, and maybe comments. So finally, stay safe and keep healthy. This is most important. Once we are safe and healthy, we can do whatever we want in life. Thank you, one and all.
Thank you very much, sir. Now I request any of the participants, if there are any questions. Participants, if you have any question, please clear your questions, please.
Sir, outset, I would like to thank Raja Reddy sir for accepting invitation. And sir, you have given a wonderful lecture in the field of drug discovery, mainly starting the natural sources of drugs, then how we plan to develop different methods to synthesize them in the laboratory, how one has to raise to the occasion, that is during the pandemic state. And you are a role model to all the delegates because during the pandemic, the drugs were not available, and if available, the cost of the drugs were too high. So your contribution to this nation is a great contribution. And we have also discussed about the different strategies to synthesize new molecules which the natural source has given us. Then methods for some of the method production of in more economical and as well as to get the better yields. So I think the lecture was more inspiring to all our delegates. And thank you, sir. Thank you for the nice lecture and a wonderful lecture all through, sir.
Thank you. Thank you so much.
Sir, good afternoon, sir. This is Dr. Balsudan, Principal Santaram College of Pharmacy, sir.
Yeah.
Yeah, thank you, sir. You added much flavor to the synthetic chemistry in the drug discovery through hard work and hard research. Thank you very much, sir, for your kind lecture and inspiration towards our researchers for betterment of the healthcare, sir. Thank you very much.
Thank you so much.
Excellent talk. We request virtual
Sir, just a moment, sir. Just a moment, sir.
Thank you. Thank you so much for the honor.
Right.
And, I'm very delighted to give my talk in this conference. I think looking forward further if any future opportunities, maybe the physical interactions will be more effective. So I'll be happy to
Yes, sir.
visit physically whenever opportunity is there. Thank you.
Yes, sir. Yes, sir. Thank you, sir. Thank you very much.